So, a very good afternoon to one and all present in this webinar. So, today we are having a renowned researcher with us, Dr. Sourav Kalra, and his topic of today's talk is overview to the computer-aided drug design methodologies and ongoing trends. So, this topic is in the in the very much trend these days, and a lot of researchers, universities, and academicians are adopting these technologies. So this will be highly beneficial to all of us and all the chemists which are connected through this webinar. And firstly, I would like to introduce our speaker, Dr. Sourav Kalra. Dr. Sourav Kalra is currently working as an assistant professor at School of Pharmacy, Chitkara University, Himachal Pradesh. He was awarded SERB-SIRE Fellowship for his research topic in silico methodologies in optimization of point of care diagnostics by SERB, and he completed his research work with the University Tecnologio de Montero, Mexico for 6 months as SERB-SIRE Fellow. Previously, he has worked as postdoc fellow, ICMR research associate at Department of Pharmaceutical Technology, Process Chemistry at Naipur Mohali, and received a ICMR RA fellowship from ICMR New Delhi. He has completed his PhD from Central University of Punjab, Batinda, in a field of pharmacogenomics, computer-aided drug design, synthesis, characterization, and biological activity of new chemical entities. He is having a very good research profile. He is having very good research and review papers into his credit with an highest impact factor of 18.03 that was published in ACS Nano, and he is having more than 30 articles, and his cumulative impact factor is approximately 165. He has more than 1180 citations with H index of 16 and I-10 index of 23. He is a recipient of various fellowship from AICT, ICMR as a SRF, ICMR RA, and SERB-SIRE. So we are very lucky to have you today in this webinar, sir. And we can proudly say that you are an alumni of ISF College of Pharmacy. So thank you very much for accepting our invitation. And now I would like to invite you for your talk, sir. So over to you, sir. Over to you, sir.
Thank you, Bhatia sir, for the kind introduction. And as you told that I am an alumni of this college, I am very happy every time when we can say invitation come from this college. I feel very proud that I am alumni of this college, and uh, I'm always present for my alma mater where I have studied, and now today. I am thankful to the faculty, to the faculty of ISF College, and to Principal Sir, GD Gupta Sir, who has invited me to give this lecture and expert lecture. As I am working in this field from the past 5 to 8 years, so I can say like whatever I have experienced in these 5 to 8 years, I am going to tell that in this expert talk. So thank you. So I am dear all faculty members, students, and my dear friends. So my topic for today talk is overview to the computer drug design methodologies and the current trends, what is going in this computer drug design. So I have been awarded a SERB-SIRE fellow where I have worked for in silico optimization of point of care diagnostics. So that was a really a wonderful experience. But we will talk about it later. And now coming to the drug discovery part. So drug discovery actually is a pipeline process which is involved in the evolution of the drugs and involves the genes to drug strategy. As we can say, like genomics, proteomics, and computerial chemistry, and all these things are a part of a drug discovery. So genes to drug strategy is followed for this drug discovery process. So firstly we will find the gene which is responsible for the particular disease, and that gene is encoding for some protein, as we have studied, like uh, uh, from a DNA replication, transcription and translation to the protein. We have studied in like our HAP. So for first we have to identify the gene, then we have to identify the protein that which is overexpressed or underexpressed, and finally we will go for the search of the drug which is required to combat that disease. And drug discovery usually starts with the analysis of the binding site, as binding site is an important part. Like it is a target in a protein. It is a target where our, we can say, our agonist, antagonist, or ligand is binding to this protein site. So the analysis of binding site is an important part for this finding this, we can say, targeting the protein, and getting one or two compounds that are active against that protein, so that we can inhibit that overexpressed protein, or we can modulate the expression of underexpressed proteins. So after finding this, we have to validate the target, that how these targets are validated. There are some methodologies like docking, and we can say pharmacophore designing, e-pharmacophore. So many of the technologies are there by which we can validate these targets. We can actually, we can say, we can isolate the proteins, and we can then we can uh, after isolating, we can target that proteins using combinatorial chemistry also. So this is the whole process. I think many of you have heard about this. This is this whole process, like finding the disease, then we are isolating the protein, and when we isolate the protein, there are three. It is involves the genomics, proteomics, and bioform statistics. Like we will find many targets. There are many targets for like cancer. If we talk about cancer, there are many targets. EGFR are there, CDKs are there, cyclin-dependent kinases, and we can say for a cancer, VEGFR kinases. So there are totally 588 kinases that are, we can say, in our body. And if many of if I say we are hardly targeting and only 5 to 7 kinases for cancer, like VEGFR, mTOR, EGFR, CDK. There are hardly we are exploiting these targets for the, but there are many more targets that can be exploited for these anti-cancer drug design. So next, we have talked about there are high throughput screening. So high throughput screening, in this we are screening up to 1 lakh 2,000 compounds for the activity against the target proteins. We are, means we can say, we have in a small test tubes, we will synthesize the compounds, and we will add protein to it, and we are screening that these compounds are active against the target. So that we can find a good, better target, and better target ligand, better ligand which is acting on that protein. So next we can use virtual screening. Virtual screening is like we are having a library of compounds, and we can means if I say there are around 1 lakh to 2 lakh or crore 10 lakh compounds, we can use this virtual screening. We can take one protein, like if I take any cancer protein, I am having 2 lakh compound or 3 lakh compound, so that it binds to the protein, and from there we can lower down to the, from 2 lakh compounds to the 10 compounds that are active against this target. So by virtual screening is an important, we can say, uh, uh, part where we can lower down this, where we can find a good target compound, good active compounds against the target. So this virtual screening is also used for the drug repurposing purposes in COVID-19. Many people are working on that drug repurposing, but that drug repurposing is done by this virtual screening methods. So, nowadays in silico models or ADME models are used by as they are replaced with preclinical testing. So many of that, like we can make ADME models for these preclinical testing from the mouse, and we can cut down the, we can say, use of mice or rats in the preclinical testing. So next, I'm going to talk about the computer-aided drug design. That is an important part. So it is a emerging discipline. and it is using several bioinformatic tools and chemoinformatic tools and computational chemistry. So it is of two types, as it is explained, like structure-based drug design and ligand-based drug design. So from a figure, it is visible. Like structure-based drug design is a design where we are having knowledge of the proteins. Like if the structure of a protein, like an EGFR is there, so EGFR in cancer, there are many drugs have been approved, EGFR, gefitinib, erlotinib. So, and these are acting on this EGFR inhibitor. So, the structure of EGFR is known to us. So, and the inhibitor is also known to us. So, we can bind, we can create new ligands or new inhibitors of this EGFR by like by library screening, or we are having some, we are synthesizing the compound in the laboratory and we want to check the EGFR, we can say EGFR score, that it is binding to the EGFR or not. EGFR score, like docking score, we want to find that that our compound is having activity against EGFR or not. So we can use this structure based technique for our strategy, for like for checking that our compound is working or our compound is active against EGFR or not. But in the second case, like ligand based drug design is done, where we can say there is no, we can say no structure of protein is available. If the structure of protein is not available, then we can use ligand based drug design technique. So in this, there we can use QSAR models, quantitative structure activity relationship, that we are having like 30 to 40 compounds which are active against some target, and we do not know the structure of that target. We can easily, we can say, predict the structure of, if we are having 12 to 30, 20 to 30 compounds or 10 to 12 compounds, we can build a QSAR model and we can predict the activity of other compounds, and we can synthesize only those compounds which have good activity against that target, which for which the structure is not known. And pharmacophore modeling, we are getting this pharmacophore modeling. In this pharmacophore modeling, we are getting some, we can say, basic features: hydrogen bonding acceptor ring, and hydrogen acceptor hydrogen bond donor, and some of these features of a compound. And we will generate this pharmacophore model, and we will screen like 2 lakh compounds or 3 lakh compounds what we are having in a database against this pharmacophore model, and get the active hits which are mimicking this pharmacophore that we have made. So, computer drug design enhances the study of the drugs and related biologically active molecules. And the target of assisted drug design is to not to find the ideal drug, but to identify the good leads that can save our experiments. So we can save time by using this technique. So these are not for, we can't find the ideal drug, but we can save our experimental time, that we are synthesizing like 20 compounds, 100 compounds in our lab. We are using so much of chemicals, chemical hazards are there. So we can lower down the risk of these things by using this computer drug design techniques. So we can, if I already have told you that many of these, this is a chart where we can say that we are using structural bioinformatics for the drug discovery. So structural bioinformatics involves all this chemoinformatics, structure-based drug design, ligand-based drug design, ADMET modeling. So we are starting with our human genome, and we are getting any one gene that is overexpressed in our body, which is coding for some protein. So from here to protein, gene to protein, there is a process of translation. So first, from in a gene, it is from a genome, gene is transcribed, and from a gene, by translation, protein is produced. On that, if the protein is overexpressed or underexpressed, we can target this protein. We can use high throughput screening, and we can use like structural bioinformatics. We can use chemoinformatics. We can use docking methodology, and we can use all the type of, we can say, techniques to from this to high throughput screening. So from high throughput screening, we will find the hit molecules. From the hit molecule, if we find some hit molecules, from that hit molecules we can find the lead molecule, that that has a potential to become a candidate drug. So from this lead, we have to do lead optimization and other things. Then only we will we have to check the activity from the animal model. We have to check that. Then only from lead candidate drug can be. This process takes so much of time. Like we have seen, like it take 30 to 40 years. But now, in if I say for the COVID, the research has been done, and we are able to find a suitable vaccine within 1 year of this 1 or 6 months to 1 year. One year, we found a potential vaccine for this COVID-19. So this drug discovery process is so much faster by using these techniques. So what is hit and what is lead? So hits are the molecules. If I say I am having a protein, EGFR protein. So if many of I am having like 30 to 40 compounds that is fitting in this cavity of a protein. So that these compounds out of 40, like 20 compounds, if they fit into this cavity, so these 20 compounds can be called as hit, and these 20 compounds can be called as hits. And if I say that only one molecule is having a good activity or good, we can say uh, good when the biological screening is carried out for this hit. So if this hit contain a good biological activity, then this is called as lead. So this lead molecule can be further taken, and lead optimization can be done. And if I say this is the active site, we are adding lead to the uh, active site. And if we add some functional groups, chloro, bromo, or any of the functional group that can accommodate in this binding site and improving the score of this compound, then this might become a lead optimized molecule. So in the lead optimization, the proteins, we are able to find that what are the active, we can say amino acids that are involved for the inhibition. What are the amino acids involved in this inhibition of this protein? And second, we will find that what are the amino acids which are we can say that expression of the protein, the changes in the protein we can find with this lead. That score, if the score is increasing or if the score is decreasing. So this is the expression of the protein, we can say. We can find these. So these are the two things we can find out this by this lead optimization. We can find it. So next are our drug designing approaches. In the drug designing approach, if I say, so there are, I have already told you that there are ligand based approaches and structure based drug designing approach. So in a ligand based approach, we are using QSAR technique. So if I say QSAR technique, so in a QSAR, firstly coming to the ligand based drug design approach. So the receptor structure of the receptor is not known to us, so that why we are using this ligand based drug design approach. So it is a by this approach we can identify the characteristics of which are common to the ligands. Like if I say I am having some quinoline type of structure, quinoline is having functional groups that are attached to it. So quinoline is common characteristic in the ligand. But if I say chloride is attached to one and bromide is attached to another molecule, so it might affect the activity of these molecules. So we can say we can having these common features we will select, and we will say that like there is a variable features like chlorobromo or any isosteric group is attached to that. So in that active set of molecules, we can use ligand based computational method in which we are having, like if I are having 13 molecules that are active against some target, so I can bring out the common features from them, that 13 ligands, and I will make equation of the activity is equal to the feature 1 plus feature 2 plus constant and so on. So activity is equal to the variable features that are present in the we can say that are present in these 13 compounds. So next we are using, so this is the mathematical equation for the QSAR method. So in the QSAR method, if I say So on in a graph, the log one by c is a we can say is a activity, and log p is a physiochemical character. Like log p, we know that what is log p. Log p is a water to ethanol coefficient, we can say, of a drug. How much it is soluble in water, how much it is soluble in octanol. So we can measure this type of features, physiochemical features, and we can predict with the activity. And we can make our features from our side, like how many nitrogens are present in the molecule, how many carbons are present in the molecule. We can, based on our, we can say, our that homo of the molecule, lumo of the molecule, based on the structural parameters, how many carbons are there, based on the number of nitrogens, how many nitrogens are there, how many oxygens are there. So these are some of the structural features of this compounds that can also be used for making this QSAR models for the we can say our compounds. So second method is the pharmacophore method, like I have here it is shown. So in this pharmacophore method, so we will take a features of the molecule. So like if there is a one ring is there, so we can mark that ring. We can, if I say this is a structure of a one steroid. So if I say there is a hydrogen bond, donor is there, hydrogen bond, donor is here. Here it is hydrogen bond acceptor. There is a hydrophobic part. So by making this, they have made a one beautiful like pharmacophore. So this they have made a distance based pharmacophore. So this is by so if we screen, we will take this pharmacophore and we will screen around one like database of compound which is having around 1000 or 2000 or lakhs of molecules. If we screen that database on this pharmacophore, we can get we can say if one to we are getting the like nearer the features. By selecting these features, we will get the molecules that are screened over them, this pharmacophore. On the basis of distance, we will get some of the molecules that are mimicking these features. So if the features mimicking features are there, so the compound might have same type of activity with this can be selected, and we can predict the activity of that compounds also using this method. So next we will go to for the structure-based drug design. So structure-based drug design is a technique which is nowadays commonly used because the number of the we can say proteins, the structural information for the proteins are expanding nowadays. There is a website called RCSP PDB. So that website I will show you. So this is the website. It is opening. So this is the database for the protein. So where I will show you that it is a PDB ID for a acyl coenzyme carboxylase, which is an enzyme of ACDD5, which is an enzyme of Mycobacterium tuberculosis. So these are also these proteins can be targeted for anti-TB drugs. So this is the site. It is having like lakhs of structures of proteins, and now this database is expanding because the many of the we can say uh, many of the proteins structures have been incorporated in that database, and with that new discoveries, so many of the proteins have been discovered. So the protein which is discovered by the scientists or the researchers, these are added to these databases. So, so we can say if we talk about structure-based design, it is a technique that accelerates the drug discovery process and using the structural information of a protein, and help to improve the lead optimization process. So, if we say that we are using structure-based design, it reduces the cost of the target identification, and we can and also target identification by, and we can say if we are filing any investigational new drug, we can file by using this as structure based drug design, because it has reduced the cost of we can say IND, investigation new drug filing. And it requires a 3D structure of an inhibitor bound to a target, and the structure obtained with the interaction with an inhibitor and the active site. of the targets are analyzed. So by this structure-based drug design, we can find that our protein is bind with the inhibitor, and that is all in the active site of that inhibitor, protein, and we can easily, we can say, find that we can that our inhibitor can have the potential against that protein or not by using this structure based technique. So structure based techniques mainly involve that docking process. So docking refers to the ability of a ligand to position itself in a active site of a protein and calculate the binding affinities. And docking algorithms can be used for the to find the ligands and binding conformation at the receptor site, and which are close to the experimentally determined structure, and also identify the multiple proteins to which a small molecule can bind. So docking algorithms are used because docking is a process in which we are having the receptor, we are having the ligand, and we are having that receptor which is already bound with some of the inhibitor that has been, with which it is co-crystallized. So what we can do, we can take that ligand out. We want to see the activity of our compound, so we will bind that our compound to that protein, and we will see that what is the binding affinity of the protein, what are the amino acids that are involved in that binding, and we can say that we will find that what are the amino acids that are responsible for it, for the we can say inhibition of the protein, and we can say that our ligand is having better activity or better score, not activity, better score than that existing ligand. Then we can say that it might be that our ligand is having this type of activity. So there are some of the docking programs like Glide, Autodock, that are freely available. Like Autodock is freely available. Glide is a with the Schrodinger, and Gold is with the CCDC. Like Cambridge Gold software is with the Cambridge, and Glide is with the Schrodinger. Autodock is a free software. And how this docking works, the docking generates a molecular surface on the protein that we can say the cavity of the receptor. I have already shown that it is shown is a blue spheres, and this is a center for the potential location for the ligand atom. So this is a, like we can say, this is the cavity. This is the cavity of a protein which is having space, so where our inhibitor or ligand can bind. So this so here in a second part I have shown that the ligand is bound in this pocket. So if we see that our ligand is, we are putting the ligand in that cavity, so it generates around 10 raised to power 4 orientations of this uh, like ligand, and only, and it gives it is on us that our software gives around 10 best conformations or 10 best orientations for the binding in this cavity. So next, if we say that next tool is virtual screening. So virtual screening is a we can say is a tool in which we can bound thousands and lakhs and many of the molecules. We can even we can take any database like PubChem is a database, PubChem Zinc database, PubChem database. There are many such databases, chemical databases. ChemBL hai, chemical biology of interest, chemicals biology of interest. So these are some of the like we can say databases that are present with us, and we can use these databases for the virtual screening. So we can so in this we are having known, in the virtual screening we can have the known structure of the organic molecules, and we can if we can also do the virtual, if I say that I am synthesizing some compound in the in my lab, and I want to see that I have only synthesized one compound in my lab, and I want to see that how this like how I want to like make many series of a compound. If I have made one, I have to make to 12, 10 to 12 compounds. So before making before synthesizing that compounds in a lab, so if I draw them virtually, and if I can check that that they have any potential for a, like this protein which I am targeting. I am suppose I am targeting some uh, cancer protein, VGFR or EGFR. If I am targeting that protein, so I will make series of 12 to 50 compounds by virtually, and I can screen out this library of 50 compounds on the particular protein, and find that which of the out of 50 how many of compounds have a good affinity towards the protein, and then only I will synthesize that that ligands with a good affinity with the protein. So out of 50 you can synthesize 10 by firstly you can virtually screen that compounds, and then you take good good activity compounds, then you synthesize that 10 compounds out of 50 compounds. So virtual screening has is a one of a good and we can say very good tool that helps in carrying out these type of studies. So in this we can calculate the affinity of a protein. We can narrow down the small number of possibilities. Like if we are having, I have already told you that we are having lack of compounds, we can narrow down it to 10. If we are having 50, like if I am having my library with me, I am having 50 compounds, I can narrow down to 5 also. I have narrowed down to 10 also. So it's our choice that how can we use this virtual screening tool. So virtual screen in a surface representation represent the docking surface and identify the region of interest. So if I say this is the this picture, it is showing like that arginine is an important, we can say here residue which is binding with the molecule tyrosine 165 is there, ASN 100 is there. So we can this is the surface representation, it has been shown. So we can find out the what are the important amino acids and what is the zone in which zone they are binding, and we can also optimize the binding score of these molecules. So why docking is important? As docking is important because it helps in the rational design. Rational design means we can uh, we can design a compound where like which amino acid we want to target, in that direction we can go by using this technique. And it is also relevance in the cellular biology, in which like when we say our body is having two proteins are interacting in our body, so that we can use this technique. The protein protein docking is there, so in which we can find out that what how these proteins are interacting with each other and with the other molecular components. So this is I have taken a case study like we are having the crystal structure of a protein, like I have already shown you on RCSB PDB. So this is the structure protein. We can use two type of techniques. We can use computational chemistry or QSAR. We can make a lead compound, or we can use a virtual screening technique, and we can go for a lead compound identification by using this structure based technique. By the computer chemistry we can say this is a QSAR type technique. So these are some of the softwares that are available. So I want to show you some one software like it is a docking software. It is a like a visualizer. There are many docking softwares, Autodoc, which are free. Second, I am having some visualizer software. Pymol. I want to talk about the Pymol, and I will show you that how it works. If we go for some of the, we can say some pictures for our publications, so we can make using these softwares, these visualizers. And this is the another some software like OpenBabel that help in the conversion of some formats that we have in SGF format. We want to convert the molecules to mol2 format or any format. So these are some of the kits that can be used. So overall, the CAD helps in molecular selection by affinity and ADME estimation. By modeling, we can using homology modeling. We can make a database, 3D structure database, small molecule database. We can go for some of the screening techniques like structure-based screening, pharmacophore-based screening, shape-based screening. We will find target like I have already told you, lead molecule identification, validation. that the compound is having druggability, that it is binding to the that our compound is binding to some protein or not, to which protein it is binding. We can validate using this CAD techniques. We can go for docking and library design. We can go for discovery process. We can do optimization like lead optimization, QSAR techniques. We can go for the molecule selection. So these are some of the I can say overall picturization of CAD, that what we can achieve with this computer drug design. So going to talk about ongoing research. Like I have started my research in 2017, 2015 with my PhD. So and in 2017 we got my first article from our group. So where we have worked on pyrimidine derivatives and EGFR inhibitors. So in this EGFR inhibitors, so I have so there are two type EGFR inhibitors. Like I have for the first I have I can say dog the molecule on a wild type of EGFR. Second, I have dogged on a mutated EGFR. So in a mutated EGFR, we can say this is the cavity is that this molecule is tilted here. So this is the these are the results we have. So in this we found our key findings are that methyl 716 is an important residue that is important for the EGFR activity. And if I say this threonine 766 and methionine 716, but the methionine 716 is an important residue. It is required for the EGFR activity, and it is like when we bind the inhibitor bind to this, so this is the key interaction for the EGFR inhibition. So cysteine 773 is a hinge residue if you see. So these are like if I say talk about EGFR is having now four generation of a drugs, like first generation erlotinib, gefitinib, and in second generation some other drugs. Third generation there are four generation drugs have been available for this EGFR inhibition that are due to the this residues cysteine like irreversible inhibitor, reversible inhibitors are due to the concept of this. So this is my PhD work I have done for the CDKs, cyclin dependent kinases, where I have find that for a CDK that an important interaction is like we have to methionine, like here methionine 85, histidine 84. So these are ASP, valine, and these are like this we can say orange color. I have already mentioned that this is the important interaction for the I can say for the all the CD in all the CDKs. This is done by this structural analysis of the structural insights of all the CDKs. It has been published in EGMC, and I think it's having good impact factor. So next we have this 1,5,6 quinazolines as a topoisomerase inhibitors, where I have we are having protein structure with us, and we have seen that it is a ATP or AMP is binding in the active site. So we have this is the picture where our AMP and our inhibitor is having the like green color is our inhibitor, and this blue color is our ATP. So by this we can see that our molecule green color is fitting in the pocket of the topoisomerase 2. So here I have this is the article in medicine research review that is having around impact factor of 14, 13 to 14. So in this we have done like I have shown the cavity of aldehyde oxidase and xanthine oxidase. We have we can say uh, we have make a difference how to differentiate this active sites of this xanthine oxidase and aldehyde oxidase. So these are the some of the differences I have highlighted in this article. That these are firstly it is inside it is aldehyde oxidase, and like pink is a xanthine oxidase, and inside it is aldehyde oxidase blue. So in the next, we have worked on E-pharmacophore technique. We have taken this camptothecin structure, and we have generated a E-pharmacophore map, and we have uh, we have made a we have cut down to three series of compounds, and I have shown all these three series of compounds in which that the third 3A compound has a good score. Like if I say it is having uh, we can say first is having good score 15C is having dog score of -6.62, and second is having 6.5351B is having -6.53, and at the 15A, this 1A compound, it is having 6.28. So we can see that the last one 3A is having good dog score, and this is a, we can say it is a lead molecule, and we have made some modification and optimized the lead, and this is published in bioorganic chemistry. So this is my highest impact factor paper, like ACS Nano. It is having around impact factor of 18.5. So this is, in this we have discussed about the amino acids that are involved in the AS2 and S protein receptor binding domain, and that how this ACE and S protein of COVID, they are binding in our body, and like causing distress, respiratory distress, and all these things. So this is my PhD thesis paper. So where we have optimized, lead optimized the structure. Here I have NF, like from fluoro I have added piperazine group here and nitro group over here. So, and improving, like I have improved its water solubility of the compound, and so that this compound get easily soluble in water and can show good activity. So this is my latest paper in which we have worked on chromium, and we have we have performed a virtual screening from the PubChem database, and only flavonoids we have taken, and this flavonoid we have identified its score, what is the score, and we have made some molecular dynamic study, and this paper is published in JBSD, Journal of Biomolecular Structure and Dynamics. So thank you all for listening my talk. If there is any questions, you can kindly ask me. I am available for the question session.
Thank you very much, sir, for the wonderful session. And you have firstly very basically discussed about structure-based drug design, ligand-based drug design. Then you have told us about pharmacore methods. Then you have given overview about QSAR, then dockings, drug discovery process, virtual screening. Then you have discussed about various docking softwares, and in the end you have uh, given us a overview of your keen research area and the success story in the field. So we are grateful to you, sir, for this wonderful lecture,
Thank you.
and your prominent. We congratulate you for your these prominent research in the field of CAD. Sir, I just want to ask one thing, sir.
Yes,
What
sir.
is the clinical reliability of these techniques, and what is the success rate in clinical therapeutics?
So if I say clinical therapeutics or clinical, if we say preclinical testing, not clinical, if we go for preclinicals, then if I say we are having a very limited amount of we can say animals with us when we are dosing them. So in that case, when our dosage form or whenever compound, if we want to test it for preclinical testing, we can limit these number of compounds. Like if we are using 50 that or 15 compounds, we can limit that compound to 3 compounds or 4 compounds based upon that like good binding affinity to the protein. So I think the in a preclinical testing, number of animals can be reduced, so that if in the later stages, if we go for clinical, it might also help reduces the toxicity of this of the compounds that we are synthesizing, and in the laboratory.
So thank you, sir. So now session is open for the audience. If anybody from the audience want to ask anything from Dr. Saurabh, sir, so you can freely ask. Okay, fine, sir. We are again thankful to you. I am thankful to the coordinator IQAC-IIC Dr. Siddharth Mehan. I am thankful to Dr. Safir Kaur ma'am, Dr. Amit Chaudhary sir, Dr. G.S. Ganti Sharma sir, and all the students for participating and carefully listening the session. And from the whole ISFCP family, we are thankful to you, Dr. Sourav sir,
Thank
for
you.
giving us time and enlightening us with this CAD technologies. So thanks a lot, sir,
Thank you.
and hoping a good collaboration with you in future.
Sure,
And
sure,
we
sir.
will definitely learn a few things from you about this QSAR and pharmacore mapping, etc.
Sure,
So thanks
sure.
a lot, sir.
Thank you, Rohit Bhatia, sir. Thank you. Thank you to all the faculty members of the ISF college and IQC head, Dr. Siddharth Main, Dr. GD Gupta, director, and Praveen Gargi, chairman. Thanks for all this, giving me opportunity for the lecture. Thanks.
Thank you, sir.
Have a nice day.
Thank you, sir.